are important in communicating different kinds of immunologically active or cells or their components. And to become successful immune reaction, it required a very efficient and quick response to be communicated all over the body. Since our immune system is working all over the body, and most of the cells, they are circulating, those some of the cells are static in certain tissues. So there needs to be communication, and this the task of communication that is very important is carried out by cytokines. And we have seen how these cytokines are, how what are their types, okay, and how they are working, their chemical structure, their how small they are, and at what small concentration they are working. So they are it is to be highlighted that they do work at very small concentration. Their dissociation constant is very low. So it must be highlighted that they are working at very low concentration, and hence they are very vital, they are very important, and biochemically they are proteins, and maybe glycosylated somewhere, that gives stability, but not they are the glycosylated moiety is not responsible for their specificity, but rather the protein structure that is responsible for binding their specific receptor. In short, also we'll take 20 to 30 seconds more to see that or to mention that these cytokines which in response to stimulus, the stimulus may be antigenic part of bacteria, virus, parasite, or some other kind of antigens. So once it the stimulus is there, it activates T cell, and it activates the cell, and cytokines are produced, and they go and bind to their specific receptor which is located on the cell, and that cell performs accordingly whatever the signal conveyed by the cytokines. So with that short introduction, where we will move towards the second part of cytokines. So the other part is the three types of cytokines we are going to see. Before directly going to interferon, some immune function, one or two slides I will explain on how cytokines or what is the role of cytokines in immune reaction. Though we have seen last time that cytokines not only they have the role in activating immunity, they have a role in right from development, metabolism to embryogenesis and so on. One slide we have already seen that how they communicate between two or variety of, or there is a web of reactions how they carry out. But two more important functions that remain in last lecture, I'll just go through within a minute or so. So cytokines role in immune function, the other part that is remained, that they are responsible for B and T cell maturation and differentiation into effector. We all know that B cells do match in bone marrow itself. Rather, that's why they are called as B cells. And and thymocytes, they have to go to for a kind of what is called as training, or say maturation in thymus. This particular migration is brought about by kind of chemokines, and as we have already seen, chemokines are also the subpopulations of the cytokines. So this migration of cells to thymus is activated by chemokines. Certain cytokines mentioned here are IL-2, interleukin 2, interleukin 4, interleukin 5 are actually required for proliferation and differentiation of B cells. It's not only T cells migration, the cytokines do have role in immune system, but they also perform role of maturation and differentiation of these B cells during activation of B cells with type 2 TI and TD antigens. As we know, T dependent and T independent antigens activation is there. So the IL-2 and IL-4 have their role there. Other cytokines like interferon, TGF, you might not have heard about. TGF is a transformation growth factor. Beta, it is also a cytokine. IL-4, IL-2, and so on. They are actually required for class switching. For class switching means from IgM to IgG, this particular class switch for against a particular. So antigen remains constant for what the antigen is, antibody response is. But if the same again the same antigen, if class switch is to be occur, it is brought about by these antigens. So class switching is important mechanism in immune regulation, and IL-2 is also required for proliferation of T cell or activation of T cell. Also, cytokines have role in apoptosis. This is very interesting. Say, for example, against any particular virus or bacterial cell, cytokines have provoked immune response, and they will keep on producing. That means that antigen have stimulated, and that T cell may have producing cytokines. That doesn't mean that it will keep on producing cytokines. As we know, cytokine is a very vital component. It works as very small concentration. So its synthesis is to be stopped. So that particular cell is to be programmed for death as well. So this particular task is also carried out by cytokines. So after, as I said, as I explained, I just read out what I explained. After successful immune response, when pathogens are eliminated, they are removed from the host body. Those activated participating cells have to be removed. Now, what they don't have to keep their task continued, because the task is over. The immune response that has been provoked is successful. The pathogen has been removed. Then that time, the cell that is involved is to be removed, since continuous production of cytokines may give rise to autoimmune conditions as well as it is wastage. So that cell has to be stopped. That cell has to be programmed for death. So that is also conveyed by certain kinds of cytokines. So the two systems participating such programmed death or inducing death, that is called as TNF, tumor necrosis factor, oblique TNFR. It is a common practice to write their receptor along with. So FAS and FAS legend, this particular what is called as system or in cytokines. FAS you might not have heard about, surface death factor, and its legend that when it get activated, it will induce death. So it is interesting. The another part is again, so TNF and TNF family, they are involved in signaling activation induced cell death, apoptosis we call this, like that. Another interesting feature is that the receptors with legend gives both proliferation as well as apoptosis. How can this possible? Because as we know, TNF is also responsible for certain proliferating mechanisms, but the receptor they are binding, if it is P55, TNF receptor T55, its signal is cytotoxicity. It kills the cells. If TNF binds to the another receptor protein factor 75, it signals proliferation. So this is all about functioning, signaling of cytokines. So these are very interesting phenomena taking place. Cytokines do perform the role of communication. Okay, so let's come to today's main topic. That is that as we are going through cytokines, three kinds of cytokines: interferon, TNF, and the other one. Okay, so TNF is, as we know, TNF and the interleukins. Okay, interferon, interleukin, and TNF. Today we are going to discuss this. Let us see interferon. As this is the most main topic of this lecture. Three cytokines we are going to discuss: interleukins, interferon, and TNF, tumor necrosis factor. First, let us see interferon. Why I have selected first interferon? This is very important and simple to understand, and examination point of view, and even in other exams, your competitive exams, you find the questions on interferon and certain interleukin section. Originally, interferons are identified as antiviral agents, though now they are classified as cytokines, and we know the story behind it. So in late 1950s, 56 or 58 or so, two researcher groups at different, distantly located, almost opposite part of the on the planet, they are working on the same area. At the same time, they are working, and they have almost simultaneously reported this kind of cytokine. Of course, in different purpose or aim. One group that is working in Japan, another is London. The story behind that is that the first group, Nagano and Kojima, they are working in Japan on smallpox virus, and they are very much busy to, or they wanted to produce immunity against smallpox. So they have used UV irradiated inactivated viruses and growing them in testes, tissue culture, or skin of the experimental animals. They found that a very interesting observation they have noted, rather, that the attenuated virus, when inoculated in the vicinity of the cells, the rest of the cells, they become immune. They cannot be acted, they cannot be attacked by the virus. Similar kind of observation is noted by Isaac and Ledenman in London, but they are rather working with influenza virus, and they have hit inactivated this virus and growing in cheek choriodontic membrane. Similarly, they have seen that the adjacent cells or the area, they observe local immunity. The virus cannot multiply, infect, enter in adjacent cells. So they said both of them have might have thought that the cells which are infected might have produced something and interfered the growth or the multiplication or the replication of the virus, and they Isaacs and Lidenman, they gave this name interferon because they thought that they are interfering with the replication of the virus to the molecule. So this is how the name is given. So they gave the name interferon because of its ability to interfere with viral replication. Since interferon acts as a very low concentration, again, it took two and a half decades to produce it in sufficient quantities, as we know, for biochemical and certain biophysics kind of aspects. If you have to study of the molecule, you require to have that molecule in sufficient quantity. So it took almost a couple of decades to have the molecule in sufficient amount, and then people studied. Then they studied with the help of certain biochemical techniques and certain biophysical aspects of the structure, especially the crystallography. And Isaacs and Lidenman came up with several publications on that, and saying that this how they are, what is their structure. And later it was elucidated that the elucidation was performed, how exactly they interfere. We will coming to it, how exactly they work. So let's move. Sawan sir, I am audible, no? Sir? Hello?
Yes, sir, yes, you are audible.
Hello? Hello? Okay, okay. Because your voice is breaking.
Sir,
And clearly,
due to poor connectivity, I am. Yes, yes.
Oh, okay.
Maybe my voice will be breaking.
Okay, okay. But my voice is clearly
yes,
audible.
Yes, sir. Yes, sir. Your audio is clear, sir.
Okay, fine. So we have seen that the story. Yes, sir. Hello.
Sir, just go ahead, sir.
Okay, okay, fine. Okay. So we have seen with the help of crystallographic and certain biochemicals study aspects, they have characterized. So these cytokines, interferon, they are responsible for many variety of immunological actions, such as macrophage activation, augmentation of neutrophils. The neutrophils are required to be augmented there, and monocyte function. There are actually three types of cytokines. I will be coming to it later. In 2003, the third type was recognized and typed. But let us see chronologically. That is why in this table I have mentioned two types. So though it is mentioned that there are two types, actually there are three types of interferons. But let us see, there are two major types of interferon, type 1 and type 2. Type 1 can be again divided into two subgroups. And type 1 interferon, type 1 interferon consists of two subgroups, alpha and beta, and they are produced by leukocytes and fibroblasts respectively. And the main function of that there is, of course, antiviral activity they offer, and beta interferon macrophage activation. The type 2, that is gamma interferon, is activated. The major source is activated T cells, and the important function they carry out is macrophage activation and MSC expressions on cell, so that more and more antigen is presented to the specific cell. Okay, so type 1 interferon, let's see. It composed of interferon alpha, a family of around 20 different types or related proteins, and interferon beta. Okay, and they are secreted by, as we have already seen, secreted by macrophages, dendritic disease, and certain nucleated virus infected cells. Okay, and interferons alpha and beta are secreted by, of course, virus infected cells after recognition of viral antigens by PRRs. As we know, PRRs role is very important. Pattern recognizing receptors which are present on the or in the inside the cell of the host. So pattern recognizing receptors are the one that are going to recognize virus, and then cytokines are produced in response to that. So PRRs that are present on or in the host cell, and these PRRs interaction, or they interact with viral nucleic acid, or viral that product, or viral nucleic especially RNA, DNA, and or endocytosed viral particles. Here the actual action is the secreted type 1 interferon. So how we have seen how and when the interferons will be secreted. Okay, but this secreted activated or secreted type 1 interferons in turn interact with membrane bound interferon receptors of many cells on their surface, and the result induce ribonuclease activity. Purposely I am showing this slide again. PRR recognizes viral particle, get active and activated, produces type 1 interferon. This type 1 interferon that is produced due to PRR's interaction with the antigen in turn interact with membrane bound interferon receptors. So these interferon will bind to the receptor, specific receptor of many cells and their surface, which are present inside or on the cell, and result, and they produce a kind of enzyme, ribonuclease, that is produced. This ribonuclease has very specific activity against the here it is important to note that this ribonuclease activity will stop both the host, that means infected, as well as viral replication. Actually, it is not required to say that if host RNA, ribonuclease activity is acted upon, or if ribonuclease acts on host cell, it will not provide the protein synthesizing machinery for the viral replication. So these products will interfere with viral replication, as well as it stops the cellular protein synthesis. Okay, so host as well as viral replication will be halted. Thus, it interferes with viral replication. Okay, so it interferes with virus infected cells from replication and making new viral. It stops making new or production of new viral particles. But again, note that but also destroy cells. Okay. So type 1 interferons are, let's see its structure as we are talking about crystallography and all. These are dimers of around 20 kilodaltons polypeptides, and mostly they are helical in structure, and many members are glycosylated. As again, we have seen that glycosylation is offering the stability to the structure and not specificity. So type 1 interferons are, as we have seen, they are antiviral action. They are used in treatment of several viral diseases like hepatitis. Type 2, let us see. So which is otherwise known as interferon gamma, is produced by activated T cells and NK cells, and is secreted as a dimer again. It is potent modulator of adaptive immune response rather. So uh, that biasing, that shifts the immune response towards CMI from rather than antibody response. And that is why it has been said biasing. T cells head towards TH1 type that induces activation of macrophages and destruction of intracellular pathogens. And that's why differentiation of cytotoxic T cells. So it is shifting or we can say it is modulating a particular immune reaction towards this side. Okay, activation of macrophages. So all the interferons increases. All these are those are involved in increasing the expression of MHC complex, and hence presentation of that particular antigen will be increased, and more immune reaction will be provoked. So the other one, the interferon gamma, is used to treat diseases such as leprosy and toxoplasmosis. As we know, in both the cases, CMI is reported to have a good prognosis rather than antibody-mediated response rather than HI. So, and as in early stage, in previous slide, we have seen it bias or it shifts immune response towards CMI. And that is why interferon gamma is used to treat such diseases where antibody response are known to have less effective in prognosis, in healing and all. So again, the term bias, the adaptive responses towards cytotoxic response towards CMI. And minority members of this family also contains some interleukin molecules. From their name, IL-10 seems to be like interleukin. They shared a very identical or similar structures with that of gamma interferon, and produced by this particular cytokine is produced by T, B, and dendritic cells that regulate immune response in turn. Okay. As I have mentioned, there is a third class of third type of interferon, though in table I have mentioned only two, called interferon gamma. Actually, this is the subtype of this gamma itself, or type 3 interferon family. As I said, it was discovered recently, comparatively that recently, means still it was in 2003. We cannot now say it is recently, but compared to that of 1950s, it is recently. So there are three members of this family: interferon gamma 1, gamma 2, and gamma 3, and their cytokine names are IL-29, 28, and 28B. IL-28, IL-28B, and IL-29. These are their names. What they do, or how they are important in immunological activation. They have the action as that are similar that of the type 1 interferons. Okay, type 3 interferons upregulate the genes that control viral and cellular growth. As we know, they inhibit cellular ribonuclease activity, and that is why the adjacent cell get also get induction of or resistance. Crystallography I have mentioned. This is a figure from 2B. I must express its courtesy that I have taken this figure from the book 2B. Here you can see this is what the interferon, and the yellow and green are the receptor molecules. As also text at the bottom, the interferon and ligand binding chain of its receptor. It is X-ray crystallography analysis of interferon. You can see dark purple color gamma interferon bound to its ligand, okay, alpha and beta chains of receptor. Okay, once this binding takes place, the cells to which these receptors are, the cells that are displaying these receptor will get activated and produce, for example, here ribonuclease that will stop viral replication. In this case, for example, okay. So this is all about interferons structure and all. Some other information of interferon is also interesting to note that interferon gamma is used to treat diseases, as we have already said. Okay, so this is all about interferon. Now let's move to interleukins, another kind of cytokine, most commonly encountered name. The students, the beginners, especially whenever the term cytokine comes, you see some one or other IL, interleukin. In the first lecture, in introductory lecture, we have seen the nomenclature of cytokines, how differently it was called, and finally, in 1971, in the workshop, the cytokine, the universally accepted name is, the term has been accepted, though the terms like lymphokine or monokine, the cell city synthesing, is still stuck up with the. But okay, this is the another kind of interleukin. This is another kind of cytokine, that is interleukins. Why I'm emphasizing on its name? Because these are the kinds of cytokines the beginner in immunology or microbiology or biotechnology they came across many times, that this particular interleukin has activated this particular macrophage or that particular B cell, or differentiation or immunological activation and cascade reaction and all. So interleukins are most commonly pronounced or found cytokines in many of the research papers and many of the immunological reactions, though they are not the only. Rest of the cytokines types, they are also equally important. So intermittently I keep on saying that what is the function of cytokine? They are the molecules that communicate between two different cells. They communicate different signal. These are basically signaling molecules, and communication is their function, so that students will be aware of that. So we have seen one cytokine type, that is interferon. The another interleukin we will see now. In its introduction, originally recognized as the leukocyte activator factor, as we have already seen, leukocyte activator factor, it was called in nomenclature we have seen, and as B cell activator factor later, and finally it was named as Inter1, or the type interleukin basically. So cytokines named as interleukin. What is it chemically? It is important to see. Again, I'll mention that all the cytokines, they do act at very, very low concentration. So their production and their study becomes becomes difficult. So chemical, what's the structure of it? It is again a soluble polypeptide. We have already seen in introduction what cytokines are. They are most of the cytokines, they are soluble, they are free, they are moving, but some of them are stuck up with the cell as well. So what are interleukins? Their structure and their so these are soluble polypeptides occurring in two molecular forms, alpha and beta, mainly produced by monocytes and certain other nucleated cells, macrophages, T cells, and all. Its generation is, or its production is stimulated by, of course, stimulator is antigen, toxin, injuries, and since they can be stimulated, can inhibited as well, and that is why most of the inflammatory reactions, our physician prescribes us certain steroids. Okay, so they are inhibited by corticosteroids, certain hormonal, certain molecules, inhibitors like cyclosporin A, prostaglandins, but mostly steroids, even simply other kinds of steroids as well. So they could be stimulated. Their production could be stimulated. Their production could be inhibited as well by certain synthetic molecules also. Okay. So let us see immunological effects of interleukin. What they do in immune activation. As you can see at the top, stimulation of T cells is the very important function that is carried out by interleukin. Again, interleukins are cytokines, and cytokines do communicate to different cells. So it's important to see how T cell talks to B cell. So that the language is coded, decoded by cytokines. So they what the immunological effects of interleukin 1? The stimulation of T cells for the another kind of cytokine, that is interleukin 2, and so many others as well. So this is a very simple one task that interleukin 1 carrying out. Also, B cell proliferation and antibody production, neutrophil chemotaxis, and phagocytosis. B cell proliferation, as you know, B cell is converted into plasma cell, and plasma cell will produce antibody. This particular differentiation or this particular proliferation is accelerated by interleukins. As you know, in inflammation, there needs to be more and more neutrophils accumulated. This is also brought about by interleukin 1. Okay, and phagocytosis as well. It also mediates different hematological, physiological, metabolic processes, inflammatory reactions. As I mentioned just earlier, neutrophil chemotaxis at the site of infection or at the site of trauma. If we have an infection or if we have a trauma somewhere on our hand, it swells. This is because of the what is called as inflammation, and inflammation is nothing but outpouring of these neutrophils or other specialized cells to take care of the trauma or infection, and they are output through the venules. So this is brought about by this kind of interleukins again. So they affect acting on bone marrow and epithelial cells, synovial cells, fibroblasts. These are connected to osteoclasts. These are the bone tissue, bone cells. Hepatocytes, liver cells, vascular endothelium. Of course, endothelial system is very important in that, and other target actions. And that is why interleukin 1 is very important cytokine among this family, interleukin family cytokines. Okay. And last sentence, the bottom line is very important. They are also responsible for the pyrogenic effect. The fever that is produced in response to most of the infections, as fever could be considered as one of the defense, because some of the bacteria or some of the infecting or invading pathogen may be very much temperature sensitive, and they may be eliminated just by having or elevated temperatures other than 37. So this pyrogenic effect is also induced by such interleukin 1, and as fever is considered as one of the defense mechanism, or it's one of the way to let the invader, as these invaders bacteria, for example, they are sensitive. Some, not all, of course. Along with TNF, this interleukin is responsible for hematological changes. One that is shock, it's septic shock, as we know, due to infections. There may be sudden blood pressure fall, and that shock is being put. Meningeal inflammation in bacterial meningitis. So, along with other cytokines, interleukin do perform this inflammation. It's this particular kind of inflammation. And cytokine inhibitors such as dexamethasone can there is a deleterious effect. Hello. Chavan sir, am I audible?
Yes, sir. You're audible, sir.
Okay.
Yeah. That's good.
So that fever produced to eliminate certain bacteria. So along with TNF, they also stimulate, or they also bring out uh, achieve the immunological changes. Certain cytokine inhibitors, as I already mentioned, dexamethasone, corticosteroids can protect such deleterious, because meningeal inflammation, meningitis, and all that is going to create a problem. Uh, are useful. That is why you can use that steroid therapy. However, so this is one negative aspect. This seems to be fever and inflammation, but don't forget that these are one of the defense mechanisms. But over such mechanism may create a problem, and that is why we need to have such steroidal and some other um, uh, synthetic molecules to inhibit their deleterious effect. Sometimes, of course. Though these are the some deleterious effect, IL-1 has helpful effect in many severe infections. They do activate immunological reactions against bacteria and viral or invaders, and especially their activation is very important in immunocompromised persons. So, what is the let us see at the another kind of immunological reaction it is provoking: pro-inflammatory and other signal promoting mechanisms they carry out. So again, cytokines are basically signaling molecules. So pro-inflammatory signal. Let us see how it. So inflammation is an effect. The signal is brought about by, or is activated or initiated by these kinds of interleukins, which are the cytokines. So interleukin 1 cytokine are typically activated very early. These cytokine family members, cytokine interleukin 1 cytokine family members, generally they are activated early in the immune response by disease and monocytes and macrophages and T cells and all. So interleukin 1 production is stimulated by recognition of bacterial, viral, or parasitic antigens by innate immune response. So once there is a presence of such antigenic part of bacteria or viruses, um, the first reaction is that these cytokines get activated and produced. Uh, these cytokines are produced and they activate the specific immunologically expertise cells. So communication, basically. So interleukin 1 family members are generally pro-inflammatory, as many times we have seen neutrophils accumulation through venue. That is, they increase capillary permeability. Of course, it is required if at the site of infection, more and more defense cells, more and more military forces to be deployed there to fight with militants. It is just like, and that is why inflammation has to be there, will be there. So this particular signal that militants have arrived at particular site is brought about by the signal is brought about by uh, cytokine, and that is why they are produced early infections state, and they are responsible for this pro-inflammation. As they are pro-inflammatory, more and more neutrophils are outpoured through the venule at the site of infection, with amplification of a particular leukocyte and their migration as well to the affected tissue. So they also have systemic effect, like in livers, they produce APP, acute phase protein, and acute phase protein as INF and IL-6, etc. They are responsible for fever. So these APP, as mentioned, acute phase proteins, it's a systemic reaction produced, signal produced by this kind of interleukins. Okay, so these APP, acute phase proteins, further bring multiple protective responses, including the destruction of viral proteins, production of certain viral um, fever, so production of systemic fever. Okay, fever is to be systemic, of course, there is no need to say systemic. So production of, or induction of fever rather, it will be the proper word. So induction of fever, that may remove or that may eliminate the temperature sensitive invaders. IL-1 also trigger both T and B cells at the induction of adaptive immune response. So their induction of both the immunologically important cells is brought about by this kind of cells. There are cytokines of IL family. The other one that is IL-18, IL-33. These are some other members of the cytokine 1 family, the interleukin 1 family. And interleukin 18 is related to its action is like IL-1. And it has similar functions expressed as monocyte, and they are expressed by monocyte macrophages, disease secreted by immune response, secreted early in the immune response. On the other hand, the other member of interleukin family 1, that is IL-33, constitutively expressed in smooth and muscle and bronchial and other tissues, and expression can be induced by IL-1B or TNF, tumor necrosis factor, that is another cytokine. Okay, and IL-33 is known to induce T helper cell type 2 that would uphold T lymphocyte interaction with B cells, okay, and mast cell, eosinophils. You can imagine mast cells, eosinophils, okay. These are all responsible for allergic reactions or inflammatory reactions. In short, some other interleukins, you can see IL-3, and you can see here their main function. I will concentrate on IL-6 as it is produced by T helper cell, macrophages, and fibroblasts, and its function is B cell differentiation, okay, and immunoglobulin G production, and the APP that is responsible for fever. IL-9, it is responsible for proliferation of the cell growth, T cell growth. If a situation comes that a T cell is to be proliferated only, or their growth is to be accelerated only, IL-9 is the signal for that T cell activity. Okay. The last type of the cytokine, that is TNF. Interesting one, tumor necrosis factor. TNF family of cytokines that regulate the development of and hemostasis of cells. Acting, this is to be noted, skeletal, neuronal, and immune system, along with other tissues as well. So TNF works rather than very specialized here. And TNF related cytokines are strange in that they are mostly anchored tightly into the cell membrane. Okay, and they are transmembrane protein with their structure is that some part is transmembrane and the other part may be some part is intracytoplasmic and some is extracytoplasmic. Embedded one is the N terminal, and the protruded one is the C terminal. As it is C terminal, which is variable one, that has to be out of the cell. And some members can also be present and soluble. They circulating. And TNF occurs as two types, alpha and beta. Hence, they are also named as, most of the time, they are also mentioned as lymphotoxin alpha and lymphotoxin beta. TNF alpha and TNF beta. So basically, this is a serum factor to provoke hemorrhagic necrosis in certain tumors. So what they do, they stimulate hemorrhagic necrosis, and hence named TNF. And the same substance was also independently explained as cachexin or cachexia, as we know, loss of weight due to certain clinical conditions, especially diarrhea and all. So, they are also related to that. It is produced mainly by macrophages and monocytes, and it resembles its immunological mechanism as IL-1. And it exerts immunomodulatory effect on other cytokines. So TNF induces production of other cytokines, and that cytokines may have cascade kind of reactions. TNF beta, formerly known as lymphotoxin, mainly produced by T helper cells, and its effect is similar as that of TNF alpha. So necrotic effect and cachexia and all. Of course, as other cytokines have reaction, TNF also binds to its specific receptor and probe the particular signal. Lymphotoxin alpha is produced by activated lymphocytes and can convey many types of signals. So they have to bind their specific receptor which is present on the specific cell, or rather that receptor is displayed on the specific cell. This TNF, when binds to neutrophils and other cells like osteoclast, bone cells, they stimulate the signal and leads to increased expression of the MHC, and that is how the antigen presentation is increased, and more immune reaction could be provoked. So lymphotoxin beta is a membrane-bound cytokine and is important in lymphocyte differentiation. There are 5 physiological significant membrane-bound interferons, membrane-bound TNFs, tumor necrotic factor cytokine family. And one is already mentioned, lymphotoxin B, that we have already seen. Lymphotoxin B is a membrane-bound cytokine. But rather the other, that is BAFF, you might have heard about B cell activating factor. You may come across somewhere in the books. So that, as its name, B cell development and activator. BAFF, as I said, what the full form of it is B cell activating factor. April, and this is another interesting one. April is nothing but proliferation inducing lesion. I'll repeat, proliferation inducing lesion. Its again function is to develop and activate B cells homeostasis. Another one is CD40L lesion. That the function of what it does is that cytokine expressed on surface of the T cells, and it has to do with the T cells activation. Another one is important, FAS. Many of the times you come across this particular kind of reaction, FAS, FASL. FAS, this stands for specific factor, and it is cell death. So it is such 5 very significant membrane-bound cytokine, membrane-bound or soluble. Their active state is assembled in trimer. They are basically trimer, beta sheet structure. I will come to it later. Chavan sir, I'm audible, no? Hello?
Yes, sir. Yes, sir.
Okay. Some message problem. Anyway, so they assemble in the trimer, beta sheet structure it has got. We will come to its structure. And mostly they appear in either homometric or heterometric. So the 3 arms, they may be similar or they may be different. So homometric or heterometric they may be, and do occur between lymphotoxin alpha and lymphotoxin beta and April and BAFF like. Their crystallography study have revealed that TNF family member cytokines have a certain conserved tertiary structure and fold into a beta sheet structure, as we will come to it. Actually, these conserved residues direct the folding in internal beta strands, in turn produce the trimer formation, and that trimer formation offers particular conformation to it. This is what it's the TNF family member act as in vivo. It is important to know. So this is the structure of that TNF family member. So we will stop here. These are my references to mention. Okay, Sudha Gamble, Kobi, Anand Narayan, David Mel. So thank you very much. This is the last session of my interaction with students on countrywide classroom teaching program organized by Digambar Rao Bindu College, Bhokar. I'm very much thankful to organizers and college that is running a very good activity. Thank you so much to one and all, Dr. Chauhan and his team, and all the listening students and the students that are also listening and observing and watching on live YouTube link. Thank you so much. Over to Chavan, sir.
Thank you very much, sir. Thank you for a nice presentation on the cytokines, and consistently you have given us time for this countrywide classroom teaching program. Now it is requested to all the students, if they have any queries over this topic, like cytokines, they can ask, sir. Either I think that the current situation, that is COVID-19 pandemic, somewhere it have some relation with the cytokines, because these are the receptors, those are involved in the entry of these different viruses inside our cell. So if I am right, sir, they
Yes.
have yes, they have some involvement there.
Yes, in last lecture I have mentioned that
Yes,
cytokine storm, that is
yes.
a complication of COVID-19. So the virus
Mm-hmm.
triggers certain cytokines, and due to their inflammatory reaction and deleterious effects produced at the vital tissues like lungs and heart and some other area, that is rather
Mm-hmm.
creating a problem, and because of that, that particular vital organ or vital tissue cannot function properly, and this
Mm-hmm.
cytokine storm is to be inhibited. That is
Yes, sir.
another task of the physician, rather than to also check out for antiviral medicines.
Mm-hmm.
Of course, antiviral medicines will stop, but once the cascade reaction or the cytokine reaction is induced, it becomes very important to manage that particular cytokine storm. And cytokine storm is a major killer in the present situation of COVID-19.
Correct, correct, sir. Apart from this, I think that HIV also have some implemented reaction or some involvement in entry of the HIV virus inside the cell. Either
Yes,
may
sir,
some
of course.
some type of cytokines like chemokine receptors, that
Yeah.
CCR 1 to 5, then I think that 8 and 9 and 12, and SDF. These all receptors have some what we can say, that some restriction to the HIV virus inside the cell. If we
Yes.
have the mutation apart from the wild type gene in the in chemokine receptor, they may restrict some level of some extent of restriction to the HIV virus to enter inside the cell. So such a
Yes, sir.
person who's having that chemokine receptor 5 or 32 deletion, they have more resistance for the HIV virus to enter inside the cell. Yes,
Yes, sir. That
I'm
is
right.
why sensitivity and the scenario of HIV is different, especially ARC. We call it as
Mm-hmm.
AIDS-related complexes. So before
Mm-hmm.
AIDS, there is a phenomenon. I mean, it could be divided into through GPL, ARC, and then AIDS. So
Mm-hmm.
first stage is asymptomatic. Second is GPL, that is
Mm-hmm.
generalized persistent lymphadenopathy. So
Mm-hmm. Right.
lymphadenopathy is because all the lymph nodes are affected, since virus directly infect T cell, and
Mm-hmm.
that is why there is a load on lymph node, and lymph node
Mm-hmm.
inflame. Okay, so that is
Right.
called as generalized lymphadenopathy, and then comes the ARC. So here, that is why ARC scenario is different depending upon the racial immunity against the virus. But the problem is that since virus is directly attacking lymphocytes,
Mm-hmm.
and their number continuously go on decreasing, and even it comes to as low as 5, 7, 9. So it has to
Mm-hmm.
be around 25 or so.
Mm-hmm.
33, and that is a problem. Though there is a cytokine, they are modulating, they are conveying the signal to produce more and more antibodies against it, but T lymphocyte, that is a major fighting element, that is
Mm-hmm.
going on decreasing. That is the main problem in HIV, and that
Mm-hmm.
is why we are difficult, or that is rather not manageable, sir.
Yes, sir. Yes, sir. Yes, sir. I think that in some of the Indian and African population, we have the lesser amount of mutant cytokine genes, and
Yes, sir.
that, yeah, that we have a lesser amount of resistance toward the HIV. But in some of the Caucasian population, like what we can say, that it is a white population, they have a
Mhm.
larger number of deletion in such kind of gene. So
Yes.
they have a more resistance toward the HIV.
Yes,
Hopefully,
sir.
yes. Yes, sir. We have a truthful discussion and lectures in the countrywide classroom teaching from your side, and for our student also, they have a get a better knowledge of that immunology topic, subject immunology, which is benefited for the microbiology as well as the zoology student. Definitely, our your lectures enhances our student knowledge about the immunology topic. Definitely, sir. If you have some spend of time in future, then we also request you to get some one another lecture on such a relevant and important topic for both all the life science students.
Sure, sir. Sure.
Hopefully, yes, hopefully in the May, sir, as we have a packed schedule in April. But if you have a time for us, then kindly give us one more lecture on in the topic of either in immunology or if possible come across with the topic of stem cells.
Sure, sir. It's my
Yes, sir,
pleasure.
definitely. Yes, sir. Yes, sir.
Thank you. Thank you, Arjun.
Once
Thank you, one
again,
and all.
yes, sir. Once again, I thanks all the participants for attending this countrywide classroom teaching program for today's session, and hope to see you again in a noon session at 2 o'clock, where we have Dr. Prashadjit Beli, sir, and for the discussion today. And so I once again thank you very much, sir, for joining with us,
Thank you.
and all the participants. For now, I declare that this session is end here, and we will be tuned at sharp 2 o'clock in the noon. Thank you very much.
Thank you, sir.
Yeah. किती आवाज गेला मी तुला कल्ला केला सर्व गेला आता चालू होतं म्हटलं उद्यापासून सांभाळायचं आता